like just an introductory or enrichment video, but
Yeah.
then you have to provide some maybe questions, and then there will be like a Q&A, so answers. The students would have to provide you answers. No? So maybe for the SUCs, we have to guide the teachers. Maybe you can start it in NNSU and EFSU also out there.
Yeah.
Yeah.
RITM, we're going to join daw, ma'am, RITM, sabi ni Mad Standard.
Yeah. I know she, director, Carlos, Cecilia, yeah, she also vibered me. Yes, it's great. Good morning, good evening, ladies and gentlemen. Welcome to the PAASE for Philippine Innovation Fireside Chat Series. I'm Gisela Concepcion of PAASE. I'm a PAASE president and an executive member of the National Innovation Council. So today, uh, we welcome a very special, distinguished speaker. He's Dr. Edsel Salvana, Director and Professor of the Institute of Molecular Biology and Biotechnology of the National Institutes of Health, University of the Philippines, Manila. So many of you might recall that Dr. Edsel Salvana was a very prominent medical figure during the pandemic, and he became one of the important consultants of the I forgot the name of that, interagency uh, coordinating team to manage the pandemic. We still have a few participants, but you must know that we invite everyone here in the scientific community, and that would be the National Academy of Science and Technology, the National Research Council of the Philippines, the Department of Science and Technology, and I also shared it with the National Innovation Council Secretariat. And I think one of the reasons we're holding this is because PAASE is one of the stakeholders in innovation. And I think soon you may receive notice of the launch of the innovation grants or the innovation competitions. So aside from that, there is value to this series of fireside chats because we cover all areas of R&D and innovation, where innovation would be at a higher technology level, TRL. And I think that there's a lot that our students in the universities could learn from the videos that we have posted on our PAASE website. So, well, please let everyone in your universities or research institutes know that these videos are available as a MOOC, massive online learning course to be used for free by any university or institution. And they would be introductory to a particular subject or topic that you would be teaching. And you could then use it as an enrichment video as well, and maybe provide some questions to your students or to your researchers that they would have to answer as part of the requirements for the course. So I'm just still on the introductory part because we're hoping that more participants are able to join us. But at this point, I would like to yield the floor to my distinguished co-host. This is Dr. Alvin Colaba, Distinguished Professor of Mechanical Engineering at De La Salle University, a past PAASE president and the current vice president of the National Academy of Science and Engineering. Ladies and gentlemen, Dr. Alvin Colaba.
Thank you, Giselle. Good morning again, everyone, and good evening in some other parts of the world. Yes, Giselle, this is our 17th edition of our Fireside Chat on Philippine innovation. And we are very, very excited to have another distinguished speaker for this Friday morning. Edsel Maurice Salvana is a professor of medicine and infectious diseases and is the director of the Institute of Molecular Biology and Biotechnology at the National Institutes of Health at the University of the Philippines. Also an adjunct faculty for global health at the University of Pittsburgh in the US. He is also the special advisor for HIV and AIDS to the Secretary of Health. In 2017, he was chosen as a TED Fellow for his work on HIV and awarded a Senior TED Fellowship in 2020 for his work on restoring vaccine confidence and improving science communication. He was conferred the Order of Lapu-Lapu, rank of Magalong, by the President of the Philippines for his contributions as a senior government advisor during the COVID-19 pandemic. His work on HIV, molecular epidemiology, and diagnostics has won numerous awards, including Innovation of the Year from the Infectious Diseases Society of America Foundation and the Eduardo Quisumbing Medal from the National Academy of Science and Technology of the DOST Philippines. Dr. Salvana writes a weekly column entitled Clinical Matters for the Manila Bulletin, which offers scientific perspectives on the COVID-19 pandemic and other diseases. Ladies and gentlemen, it's my honor and pleasure, and of course for PAASE, to have Dr. Edsel Salvana for our guest speaker for today. Edsel?
Thank you very much, Sir Alvin. Thank you then to Ma'am Gisela for inviting me to do this webinar. And we're really very excited because for the first time at ID Week, which is the Infectious Disease Society of America joint meeting with a bunch of others like Society for Healthcare Technology in America, Pediatric Infectious Disease Society, the Pharmacists Infectious Disease Society, an international innovation, one innovation of the year. And this was last October at in Boston. And so I'll tell you a little bit about the innovation itself and also discuss future steps. So I'm just going to share my screen here. All right, so good morning and good evening to everyone on both sides of the world. And we're going to talk about this portable HIV drug resistance testing in resource-limited settings, which we developed with funding from the Department of Science and Technology from the Philippine Council for Health Research and Development. So the problem really is this. 1 in 10 newly diagnosed people living with HIV is infected with a drug-resistant virus. Now, if you were talking about bacteria, you may have cultures, and we can figure that out. For HIV, it's a lot more difficult because you actually have to do sequencing. So few low-income countries are able to perform HIV drug resistance testing. And so this is a problem because if I tell you, say it's a bacteria, and I say 10% of them are resistant to something, then you'll want a culture, di ba? Sa virus, particularly HIV, it's not as easy to figure that one out. So resistance testing for HIV is highly technical and it's expensive and you require expensive equipment. And really, there's really only 2 laboratories in the Philippines that are doing HIV drug resistance testing, RITM and our lab, and it takes about 2 to 4 weeks, sometimes longer, to transmit results. In fact, before we started doing it in NIH, it was really only RITM that was doing it, and they got overloaded, so it took a while. And then Raul Destura, who was the director of the IMBB at that time, got tired of hearing me complaining all the time. So he said, why don't you set it up? And so that's actually what we ended up doing. And so we're one of 2 laboratories in the Philippines that does HIV drug resistance testing. So low-income countries also have limited antiretroviral regimens. For those who are not so familiar with antiretrovirals, we typically use 3 drugs at a time because HIV mutates very fast. 2 drugs from the NRTI class and 1 other drug either from the NNRTI, PI, or INSTI class. And because there's not much, a lot of the regimens cost a lot of money, the ARVs that are available in resource-limited settings are actually quite limited. So it's kind of a double whammy. You can't test na nga for resistance, tapos kulang pa yung gamot mo that you can use to treat HIV. So we know that resistance really increases the risk of treatment failure and death in our HIV patients. If you look at this map, you can see surveys for HIV drug resistance. So one of the first questions is, why are there no surveys in the US? Why are there no surveys in Canada or most of Europe and also Australia. And the reason for this is they don't do surveys there because you do a baseline HIV drug resistance test for every single patient at the start of treatment. Those other countries don't because it's expensive. And so you have to do surveys just to kind of get an idea of what the resistance is like. And these surveys are difficult to do. They're few and far in between. And so you're really only getting a snapshot of maybe 5 years ago. And that's really not an ideal situation. Whereas everybody in the US, when they get enrolled into treatment, they get a drug resistance test at baseline. And if they fail treatment, they get a drug resistance test as well. Otherwise, you're really flying blind. And you can see that they've looked at a lot of these in Africa and also in South America. And if you look at the rates, it's kind of scary because, like, for instance, for efavirenz, which is part of the first line regimen from a few years ago, the prevalence of pre-treatment, this is before starting treatment, drug resistance is as high as 15% in some parts of the world. And so you're not testing these people, you're starting them on a regimen that is doomed to fail. And what ends up happening is they can transmit that virus to other people, to their other partners who will be infected with drug-resistant virus. So you see that there's a really big problem both ways. First is that you don't have the capacity to test resistance at baseline, even as resistance rates are going up. And second is, in the poorest countries, we have limited antiretrovirals to offer our patients, and we don't even know if they work at the very start. So this is a problem that's kept a lot of people up, including myself, and this is why we thought about maybe developing a test just like this. So the potential solution is you want an HIV drug resistance test that is portable, because the problem is, since this is sequencing, you usually need a full lab. And kind of the funny aside for this is that when we developed this test, we were saying, well, you still need at least a PCR lab to be able to do this sequencing with a portable sequencer, the MinION. And so we were worried how many places in the provinces have PCR. But then, with the COVID pandemic, now everybody and their mother has an RT-PCR in their lab. So actually, this is really a good time to bring out this technology, because all you need is that simple PCR, and people can start sequencing off those amplicons. So the test itself, using the MinION, and we've really tried to optimize it, it can be performed in a few hours at half the cost of conventional testing, especially if you can batch them. You have intake from the clinic of a few patients, and then you can just do it if you have to at that time. And so you can deploy this in basic PCR laboratories that were set up during the pandemic. So these are really the specific aims for the grant that funded this. This is actually part 3 of a bunch of studies that we did. The first was actually just drug resistance at in treatment experienced patients. The second part was baseline pre-treatment drug resistance, which actually was about 11.7% in the Philippines. And the third is really the development of the diagnostic. So we wanted to clinically validate a near point of care nucleic acid-based assay that can detect NRTIs, NNRTIs, PIs. And later on, we found we needed to put INSTIs in. So integrase inhibitors, in the last few years, we've shifted from NNRTI-based regimens to INSTI inhibitors. And the thing is, we don't normally sequence INSTIs. And now the new commercial kits are starting to sequence INSTIs, but when we developed this test, we decided to just include INSTIs anyway. So it was actually a very good development, because now, first line, even WHO says that we should be using dolutegravir, which is an integrase inhibitor, as first line. And we really want to try to deploy the mobile sequencing platform to off-site locations. Again, there's only 2 labs in the Philippines that does drug resistance test. We get blood sent to us from Cebu, from Davao, and it takes a while to get those results back to them. With this test, we can try to set up those sequencing facilities. We can give them technical assistance, and at some point, they're going to be able to generate their own drug resistance reports for patients who are about to start treatment and for people who are who are failing treatment. So this is really just the workflow. This really entails a 2,800 base pair amplicon, which actually spans the whole gene. That it includes the PR, the protease gene, the reverse transcriptase gene, and the INSTI gene, all on the Pol fragment. And so the amplicon undergoes nanopore sequencing by MinION. And then we do bioinformatics and figure out the resistance mutations. Report is generated and sent to the clinician. And really, this includes RT-PR. INSTI, we get a good chunk of it, but we're still trying to optimize it so that it gets the last part. Although most of the important genes in the first part are actually sequenced. Although we are trying to optimize how to get that last part in. And the cost is, less than half of that of Sanger protocols if you optimize it. Oops, sorry. So I'm sorry, it's a busy slide, but these are just really the costs that we're thinking about. In the past, it cost about 5,000 pesos, and that's not including INSTI. For Sanger actually, and if you include INSTI, then it goes up to about 8,000 pesos. If you do NGS, it's really expensive. And for the MinION, we can get it with INSTI about 4 or 5, probably even less that if we batch. And the sensitivity and specificity when we did samples from our biobank really showed very good concordance with Sanger, which is still considered the reference standard at this point. And it's actually more sensitive and specific than Sanger when you use a composite gold standard, because we know that the mutations that you detect are probably real, and that if you don't detect any mutations, that's probably accurate. So it's actually more sensitive and specific than Sanger. And when we ran the numbers, actually, for Sanger, it missed a lot of mutations that would have changed the baseline antiretroviral treatment. There were some that MinION missed, but a lot less than Sanger missed compared to the MinION. This is just the ladder that shows the 2,800 base pair amplicon. And that's what the MinION looks like. For those who haven't seen a MinION, it's about the size of a harmonica. And you really just attach it to your your laptop, and you can do sequencing in the field. In fact, there's one that's like the size of a cell phone that that the MinION is built into, and you can sequence from that. It's a really neat thing. And it's not just for HIV. I told Dr. Montoya that this is HIV for sequencing, but it's really also a sneaky way to capacitate laboratories to sequence pretty much anything, as long as you have the primers. You can also do metagenomics, but that's a little harder with the MinION setup. And that's really just what it looks like. So right now, what we're going to try to do is, since we've already validated this in the laboratory, PCHRD actually advised us to split up the validation into a lab validation and then a clinical trial. And so we're hoping that we're going to get funding for the clinical trial this year. And we're going to start to set up offsites in Cebu, in Davao, and then hopefully in most of the treatment hubs in the country. We got about $10,000 from the contest, and we're using that because the Sanger that we use in NIH is something that's based off the WHO dried blood spot protocol. We want to actually, and it doesn't always include INSTI. So there are commercial kits that you can use to validate. And so that's what we're going to do with the prize money that we got from the Infectious Disease Foundation. And that's actually ongoing right now. We're already ordering our kits just to give a better validation from the lab side, and then also more compelling to do the clinical trial. And that's so the potential impact of this is really, if this is the first ever validated portable HIV resistance test platform, it'll have global implications for HIV testing and treatment. Instead of having to do those cumbersome surveys, laboratories, treatment hubs can actually get real drug resistance testing done at the time of starting patients on antiretrovirals, or if they're failing treatment, then you can have a much better guidance in terms of what treatment to give them. And there is a regimen called the dual ART regimen in the United States, which uses dolutegravir and lamivudine. We don't use dual regimens in resource limited settings, because if we don't know whether there's a 184V mutation, which is a resistance mutation for lamivudine, then you're basically just doing monotherapy, which will fail very, very quickly. But if we do have a baseline drug resistance test that tells you there's no lamivudine resistance, then we can use lamivudine dolutegravir regimens and avoid the toxicity of long-term tenofovir use. So that's something that we thought about. They take it for granted in developed countries because they always have a baseline drug resistance test, but it's something that's not been an option in resource limited settings because we can't be completely confident that there is no pre-existing drug resistance, and we know this happens one out of 10 times. And this includes INSTI, which was not included in the previous commercial kits. Now we're starting to see commercial kits that include them, but they're very expensive. And so this is a way to really bring down the cost and still have better coverage, have a more accurate test than Sanger, which we do in the laboratory. So this will prevent early treatment failure because then you can detect resistance and tailor the regimens. The problem is if you start someone on a regimen that has only 2 active drugs out of the 3, then the second drug will eventually fail, and then the third drug will fail, and then you'll have a multi-drug resistant HIV, which that person can pass on to other people. So this thing about drug resistance testing has always been kind of a technological hurdle for WHO, to the point that they actually have guidelines that tell you what second line, third line regimen to give in the absence of drug resistance testing. It's really just kind of a best guess, really. But now you're actually going to have data to support those shifts and guide those shifts. And I think overall that's probably a good idea because we don't want to exacerbate the resistance problem. So this was just our little summary when we were campaigning to win that contest. And this is really the announcement where we won 2023 Idea Incubator. It was the Innovation of the Year. It was also the People's Choice Award kasi medyo dinumugre ng mga Pilipino yung voting. So that's always that's always fun. Whenever there's something social media, Filipinos almost guaranteed to win it just because people started voting like crazy. So that's a built-in advantage. So just some acknowledgements for my team and also the OST for continuing to fund our work, as well as RITM and San Lazaro, who were involved and collaborated in all these. Thank you very much.
Thank you so much, Edsel. Thank you.
Thank you, Edsel.
Before we start our Q&A part, I'd like to first acknowledge the presence of some people in the audience. We have Tito Aliga of UP ERDFI. We have academician Sean Raimundo. We have faculty from Bataan Peninsula State University, Cavite State University. We have a PAASE board member and former UP official Edna Ko. We have past president of the PAASE, Al Sarafica, members Irene Villasenor, Professor Emeritus of UP, and others that, whose affiliations are not indicated, so I might have forgotten what your affiliations are. Welcome, everyone. Thank you for being here. Our Vice President and President-elect, Marge Peña, who is in the United States. And then we have Professor of the NIMBB, Miren Santos, my dear friend, people from PCHRD and well, as I mentioned earlier, we have this recorded, and so you can let your colleagues and friends know that PAASE has this set of videos that could be used in your teaching, in your courses as a part of your MOOCs. And we have a NAS corresponding member, Vic Ilag, and we have OFW leader, who's our speaker next week, Fred Silvestre. Thank you for joining us, everyone. And I would like to thank most of all our speaker, Edsel. And Edsel, I think, yeah, you were the winner. You were popular, but then even people like RITM director Celia Carlos, I know she voted for you. That I know. Anyway, so everyone, we are aware of HIV, that it's been a very serious infection, and well, in the early decades, it was fatal, but now it's not, right? Edsel. And, it's pretty well managed by the WHO and the world, but that's, I think, not entirely true because of the development of drug resistance. So bringing a point of care diagnostic um, to the localities, to the communities in poor regions of the country and in the world potentially, I think it's a breakthrough. And as far as innovation is concerned, it addresses an unmet need, a serious unmet need, Edsel. So I just like to ask a few basic questions for the benefit of our audience, which is, well, with different backgrounds. So Edsel, maybe you can just touch a little bit on, well, the active and the latent phase of HIV stages, and then the acronyms for those drugs or inhibitors. Would then let you explain what is the mechanism of infection of the virus, and then well, maybe a basic question is, how about also diagnosing the new cases of HIV, and then as far as the methods are concerned, wow, everyone's excited about that cell phone size min ion and that portable DNA sequencer, right? And so if people are wondering, what's this acronym mini ion? What does it stand for? The engineers would be interested to know the mechanism of detection or sequencing of the min ion, right? So anyway, and so those are just my basic questions.
Yeah,
Yeah.
let me start with the last question po.
Yeah.
Ion, it was it used to be under Ion Torrent. So, and it was a mini Ion Torrent. So that's why it's called Min Ion. So it uses the same, it's a nanopore technology. Basically, you have these pores that can own a strand of DNA can pass through, and depending on what the base is, there's a change in potential, and this is detected by the machine. So it can actually sequence with the it can sequence the DNA. In this case, HIV is an RNA virus, so you have to have a reverse transcriptase step. So HIV is what we call a slow virus. It's also called a latent virus because it incorporates itself into the host genome, and it normally takes about 8 to 10 years to develop full-blown AIDS as the virus multiplies and slowly destroys your immune system. And in the past, we only started treatment because at that time, antiretrovirals or ARVs were incredibly toxic, and so people would really only start treatment, once patients had full blown AIDS. And we found that actually that was not the right approach. It's better to start antiretrovirals as soon as you diagnose someone, because then in those cases, especially with the more potent and safe regimens we have now, you can actually almost restore life expectancy. Not only that, somebody who is undetectable on the viral load, if you suppress the virus, is not able to transmit the virus. So people with undetectable viral loads can live normal lives. They won't infect their partners. They can have kids the normal way. They can do surgery. So there's really a lot that's happened with HIV. And in fact, the current treatment, which is a 3-in-1 pill, tenofovir, lamivudine, and dolutegravir, it's a one pill once a day. So it's very, very easy for people to take, and the side effects are not bad at all. And so a lot of our patients are actually doing well as long as they continue to stay on the treatment.
It's fantastic. I think the HIV story, management story, is one of the great revolutions in medicine. And the celebrated case of the basketball player Magic Johnson is not lost to us, right? So in the United States. So AZT is what that ARD was, right, at that time, the AZT triple drug combination therapy. So, so in the Philippines, I know that there was a time when Dr. Tenorio, your friend, would say that HIV is spreading again in the Philippines, and it's underreported, as it is, say, in Thailand. Is that still the situation today, Edsel, do you think?
Well, actually, Thailand's done very well. They have like 1 million HIV cases, but they've actually been maintaining that, and the new infection rates are going down. Philippines, unfortunately, after a dip during the pandemic, it's above pre-pandemic cases. It's still one of the fastest growing HIV epidemics in the world, and part of the reason for that is because of a subtype shift, and that's also part of our work. Where there are very few people who believed us that there was a difference in subtypes. There's different subtypes of HIV. There's about 10 of them, and they can also recombine because actually, interestingly enough, HIV is diploid. It has two strands of identical RNA. So if two different viruses infect one cell, then they can recombine, kind of like XBB, XBC, all those things. And we were making the case, and in fact, the first two parts that we did showed that CRF AE, which is what supplanted subtype B in the Philippines sometime in the year 2000, 2006 is when it became dominant. Our data shows that it does have higher viral loads and faster progression to AIDS, comparing both subtypes. And so one of the things that we were campaigning for is really trying to get dolutegravir as first line, because we were seeing a lot of resistance with efavirenz, especially with CRFAE. And so what you're seeing with this innovation is really just a part of the work that we've done in terms of, this was a big research program, because first we had to establish, number one, that there were higher treatment failures with AE. And then second, baseline drug resistance testing was necessary because our pre-treatment drug resistance rate was 11.7%. And then the last part was really to find a way to detect that in an economical way and a sustainable way. So all that, the shift in the subtype, is really what led the Philippines to be the fastest growing HIV epidemic in the world. And that was what we were able to discover, and that was what we were trying to address.
So, Edsel, now I understand. I appreciate the work even better as a first line or frontline research, considering the magnitude of the problem in the Philippines, exacerbated during the pandemic. And I think that if you had this portable kit deployed in different parts of the country, you might even, avert or decelerate the development of another subtype, precisely because if there are drug resistant strains, because we are not giving the right drugs, or we're not making this available to poor communities, then we know that more drug resistance is developed. At this point, I'd like to note that there are many questions in the chat box, and also the hand of Dr. Al Sarafik is raised. So I would now like to turn over the floor to Dr. Alvin Colaba. Would you like to read the question of Dr. Roland Garcia from the Medical City? And then we'll proceed with the Q&A. Thank you.
Sure.
Thank you. And then I
Sure.
understand there are other questions from Cavite State. And then afterwards, Dr. Sarafika.
Yeah.
Thank you. Thank you.
Yeah, Giselle, yes, first of all, Edsel, very, very important and insightful, presentation, especially when you talk about resource limited settings. We all experience that in all areas, not only in medical, research, but even in engineering, where I am actually in, no? I'm not in a medical field. But before I read the questions, there are quite a number in the chat box. I just have one question. I would like to, Giselle, for Edsel. I know in many like resistant testing, no, there are what lack of protocol standard protocols. So how do you manage the consistency and reliability of your results, especially in this particular area of HIV, where the subject is just also a challenging, area to address. So can you just have a quick, Edsel?
Right. So, part of the problem really is there's so many subtypes, and so the primers that you pick might also have mismatches. And we've seen that. So it's important that you continue to do the validation, and make sure as many representative subtypes are validated. Right now, if we look at the samples that we're using, most of it is really CRFAE. There's some B in there, but again, the thing is, I mean, the nice thing about sequencing is, if you have a sequence, that's it. Of course, there can be some errors, but the beauty of MinION is really that, and I think this is related to the first question, it can detect quasi-species because it is a form of deep sequencing. And depending on how deep you want to go, you can detect minority variants as low as 1%. Clinically speaking, so for Sanger sequencing, we know that minority variants will not be detected if they're less than 20% of the quasi-species. For those who are not familiar with the concept of quasi-species, HIV is a terrible, terrible proofreader. So it uses RNA, so it has no proofreading. Reverse transcriptase has no proofreading. So it's the fastest mutating organism on the planet. And so it makes a lot of errors. So over time, those errors accumulate. So you have a population of viruses with actually different sequences. At the end of 8 years, you can have as much as 5% variation among the different quasi-species compared to the founder viruses. And this is also why it's so difficult to make a vaccine against HIV. Because for context, 5% is probably the difference between a sponge and a humor or something like that. It's bad. So in terms of trying to validate and in terms of trying to detect, it's almost like MinION and deep sequencing were made for HIV precisely because you can try to detect these minority variants. There's really no strong evidence to support this, but what they say is that a 5% cutoff is ideal for detecting all the drug resistance mutations. Again, Sanger can do maybe 20%, and sometimes it still misses it. MinION can do about 5 to 10% depending on how deep you want to go. Of course, it costs more money and takes longer the deeper you go. But in general, it's really a much more accurate test.
Okay. I think, Giselle, some of the questions may have been addressed already by Edsel. Of course, questions from Roland, no?
Yeah.
Of the Medical City, and even from Kabsu, Jose Matel. Anyway, there is one question that's actually probably an interesting one from USM Yvonne Saliling. Can I read this for her? No, Sir Edsel, if a PL-HIV takes an anti-retroviral drugs or undergo antiretroviral therapy, will she can't infect her partner? Or is there still a possibility to infect her partner? Thank you very much.
Yeah, so when we start antiretrovirals, usually in about 1 to 3 months, we expect that someone is virally suppressed. if there's no resistance. If there's resistance, obviously they won't be virally suppressed. But as long as a viral load is below 200 copies, the risk of transmitting through sex is zero, basically. So, that's basically the answer. If you have good viral suppression, then what we call U equals U, undetectable viral load equals untransmissible.
Question here from corresponding member Vic Ilag. Can you please explain how nucleotide sequencing can determine drug resistance? Is this based on a database of known drug-resistant mutations? What if there is a new mutation?
Yeah, so Stanford maintains a database of drug resistance mutations where they have viruses. Actually, it's very difficult to culture HIV, so they fuse the viruses with SIV, and they actually test them against different drugs in terms of the conformations. So there is a database of known drug resistance that correlates closely with specific mutations, and in fact, they have very good informatics. You just have to put the sequence into the website, and it will give you a report that says what it's resistant to, what the mutation scores are. It is very technical, but they've actually gotten it very well to the point that as long as you have a sequence and you can put it in, then you can figure out what the patient is resistant to.
So Alvin, I just
Edsel,
like
go
to
ahead. Please have
interject
that
a bit
question.
here. Yeah, I think bottom line is well, maybe people like Vic and I would wonder how good the correlation is between the genetic mutation and the effect on its 3D structure of the expressed protein, like the integrase, okay, or the reverse transcriptase enzyme. That would be the basis for uh, explaining why it is now resistant to this small molecule drug, which also has a 3D structure, because we know that the interaction between the drug and the target enzyme, that's very specific. So I think what I think Edsel has told us is that, Stanford is so sophisticated about the bioinformatics that there is immediately a kind of a computational model that will tell you what the impact is of the mutations, or several of them, or maybe just a few of them, on the structure of the target enzyme. Okay, so anyways, I think that Alvin, there are maybe just computational, not experimental. I think that's the question of Vic, who's got the crystallographers here. So bottom line is there like crystallographic data or studies already on this? Yes.
No, it's experimental po, ma'am. Kasi they
Experimental talaga. All right.
mutation and test it, and then they can say what fold resistance is seen. So
Okay.
both computational and experimental. So if it's a new mutation, they look at the impact, and then they actually do experiments to confirm it po.
With the drug, no? Yung talagang tinitignan, biglang wala ng ano.
Yes, yeah.
Oo, yun din, no? Wonderful. So, anyways, glad to know that Stanford is also got labs that are very, very interested in HIV, which would not be their, probably not their major concern now. Anyway, Alvin, there are a few more questions before we call maybe Al. So Alvin, more questions on the chat box from our ITM. Oh, or did you get your primers? Did you design them, or did you pick them up from a database? I think there's a question here from RITM Stephen Paul Ortia.
Yeah, so we designed them, and it was not easy to optimize the conditions and everything. Yeah, so it wasn't easy. That we spent, I think, more than I think almost a year just trying to optimize everything, and then also running it on the MinION. And again, until now we're optimizing because the ends of the insti, when we do the MinION, tend to get cut off a little bit, and we're trying to tweak to see if there's anything else that we can do.
So beyond the 2,800, well, you will have to add the insti sequences. Kaya pa rin ng MinION, right?
Yes po, ma'am. Opo.
Ano yung maximum ng MinION? What's the maximum number of Yeah, the size, base pairs.
But theoretically, because it's ano naman, it's one at a time. Actually, it can I think it can get up to theoretically, I think up to 5,000. But yeah, but yun nga. So it's really just a matter of optimizing, and also the bioinformatics that you can use. They're always coming up with other solutions for cleaning up and base calling and all those.
I see. So, Joy Mariette Barayray, I think, she just wants to ask this basically, what is the limit of detection viral load of the assay?
Oh, the assay that we use, Minayon doesn't do viral loads. We use Gene Expert, or there are other kits out there. So viral load detection is about 50 copies per microliter. In some cases, 20.
So it's nice that you mentioned Gene Expert, because in the early days of the pandemic, Al South Africa and we were so involved in trying to get the Gene Expert machines used for TB. Am I right? Also used for COVID. So
Yes.
now you're using this. I think that after Vic's last question, and then maybe you can just communicate directly, we will ask Al to speak. Vic, from Vic. Sorry, Alvin, I'm just asking because I've seen this.
Yeah.
There's for the applicants, applicated. Are these done using imported kits, or can these kits be made locally? What polymerase do you use?
Yeah. Actually, we just use our standard PCR. We don't even use kits. I mean, we use our own brews, so it works just fine. Any kind of PCR protocol. Well, we optimize the conditions, but it's not. We don't use kits. We use we just order the supplies like any other lab.
Okay, that's great. So you're now doing like in bulk, right? You're just doing, yeah, it's cheaper for you. That's
We will
cool.
probably put together a kit just to
Yes.
make it more
Yeah,
standardized.
like I think Raul did for his MTech kits, right? That's
Yes.
great. Anyways, I'll, Alvin,
Al,
yeah.
can we now ask
Okay.
Al?
Okay, thank you, Giselle and Alvin for organizing, and definitely Edsel, congratulations for your success in getting the kit recognized already. My concern is definitely on the scale up commercialization and regulatory. As you know, I always talk about TRIM. Your technology has to be sound, and your regulatory and IP. Well, this guy's IP with your group at TT Video and UP Manila next. But definitely my concern is on the regulatory front, having had that experience. I was part of that A-Star ASEAN Diagnostic Hub with Sydney Yee and Jimmy Montoya 5 years ago, and we were looking at kits, low resource setting kits for detecting. At that point, the selection was malaria, TB, and dengue. Of course, malaria is already being addressed by some groups. TB was Gene Expert, already covered by the Global Fund. And so dengue was the one that was chosen, and we were ready to roll it out in 2019. I actually asked Indonesia Eichmann Institute to come over to look at Raul Distura's kit. And they said, we're going to use 100 million of those kits in Indonesia, if only we can have a license to produce it in Indonesia. And Raul hesitated. 2 months later, COVID happened. So to me, I'm very concerned about the scale up, as well as the scale up strategy, definitely. And of course, the regulatory. And in the regulatory front, clearly we were kind of challenged during COVID, and we were very much encouraged by the way HSA of Singapore did, as in meeting with the developers of their kits very early on in the pandemic. Within 72 weeks, I think, they were able to roll it out with full clinical validation. And as you mentioned, Jimmy is going to fund some of your clinical validation moving forward. My key question now is, did you have a regulatory read, either with the Philippine group, which unlikely, I must say, or actually with the ASEAN group even, or the US groups, to help you, guide you, so that when you complete your clinical study, you might have at least a regulatory submission that will allow you to have very, even though limited indications for use for diagnostic detection, But I've always wanted to incorporate, especially with the UP system, UP, with Menchit in UP Manila, to always have this, especially for medical diagnostic drug and devices, to have this regulatory guidance, if I may say so, in order for us to accelerate the commercialization and scale up of your kit. Sorry, it's a little bit complex in that question, but of course, it's always say, no, Al, we didn't see it, we didn't do it, or we would love to have done it. But definitely, I just saw actually Ted, her boss, yesterday, Secretary of DOH, and said, Al, we should meet. And I said, yes, FDA, three letters, three letters to discuss. But I'll let you go ahead and answer, Edsel. Thank you.
Yeah, thank you, sir. So that actually has always been in our horizon. In fact, when we won the award, Innovation of the Year, the US side also gives advice and is still in the process of connecting us with patent experts as well. Here locally, of course, Raul is, we work closely together, and so I'm aware of all the issues that Raul had to go through with his kits as well. So there's guidance on that end. I think that even though this is already lab validated and technically you could probably use it with a clearance, going through the whole clinical trial process will probably give it more. It will probably give it more validation, and there's a lot more confidence in using it so that we can work the operational kinks out as well. And so from my standpoint, I look at this from a holistic standpoint. If you look at the world, we really have a big problem with HIV drug resistance. So even if we think about what we're going to patent here, intellectual property, the thought process is we really want it to be used as much as possible. And this is something that we want to talk to WHO about, whether this is something that they want to adapt in resource limited settings. And, like for instance, one of my idols is Peter Hotez, who developed the Novaseq, Novavax for COVID. And even though it is patented, he licensed it for free for all the different resource limited settings. In HIV, that spirit is actually very much there. So for instance, tenofovir, lamivudine, dolutegravir regimen, which is made, dolutegravir is made by ViiV. Even though it costs $700, $1,000 a month in the US, ViiV has licensed it to resource limited settings for $50 a year. So, I think that kind of model is what I'm looking at, that if this is something that can be useful for the world, then, any kind of profit is really secondary. What's really more important is we get the technology out there so it can save lives.
So Edsel, Alvin, there are several questions in the chat box. I think we should address them because they are from our SUCs. But before that, Edsel, I wanted to say that in the case of the dengue kit of Raul, well, at that time, I saw some of the early sensitivity and specificity numbers, and they were low, and then they improved a lot over time to an acceptable level. In your case, your sensitivity and specificity numbers are very, very impressive, right? So then it went through the fairness opinion report, this dengue kit, even before I think it reached those high 90s S&S levels. Then what I understand is the DOH would not buy it, even if there was good FOR, and that's because there is a competing dengue kit from China, which is much cheaper. So in the NIC, one of the things we're trying to champion is Tatak Pinoy, which is how progressive or middle-level countries are able to protect their inventions and innovations, to give the local innovators, a little edge. Okay, it might be a little more expensive because we're just starting and the volume isn't there. But then maybe we could have a policy that would support our local inventors. So Tatak Pinoy law, that's a law, and actually PBBM mentioned it at his last SONA. So we're keeping that in mind. Okay. Now, at this point, Alvin, I'm just going to again read out the question of Jose de Matel of Cavite State U.
Yeah.
I think many of us know that the leading SUCs in the country are being asked to set up their College of Medicine and training hospitals. And I just got it from PGH Director Gap Legaspi that PGH is supposed to lead in the setting up of these training hospitals. So PGH has a very, very important role to play, as well as the UP College of Medicine. I always wonder, where are we going to get the teachers in those provincial colleges of medicines in the SUCs? Malaki na naman talaga ang role ng UP professors, who've also been the ones, providing professors to our private hospitals like TMC, the Medical City, and other hospitals. No, so Jose's question is, I'm wondering if sequencing info is also used extensively, integrated into monitoring for TB drug resistance and AMR incidents and its management in hospitals are a very important question because we know that TB is really something that HIV patients become susceptible to. Yes.
So the question is actually very timely because we're starting to use more and more nucleic acid technology and sequencing technology in detecting resistance. For instance, I mean, we always use TB culture, but that takes about 6 to 8 weeks to come back in terms of resistance testing. But for instance, for Gene Expert, we know Gene Expert has a detection kit. There's actually a secondary Gene Expert cartridge that you can use if you detect rifampicin resistance. Then you use the Gene Expert TB Plus, I think it's called, that can actually also detect resistance to other agents like isoniazid, fluoroquinolones, and glycosides and pyrazinamide and all sorts of stuff. For AMR, even though right now most hospitals are using still culture, there's a few new technologies that are out there. For instance, the BioFire can actually detect resistance genes early on. And we do have them in some of the hospitals that I work in. Right now, we're doing a study that's looking at carbapenem resistance genes in Klebsiella in PGH, and we're also using the Gene Expert to detect that. So CarbaR is a cartridge that detects carbapenem resistance enzymes. So whether it's a metallobeta-lactamase, a KPC, a VIM, an OXA. So it's very, very useful for us because from an infectious disease standpoint, these strange abbreviations mean something to us in terms of our antibiotic selection. So we are seeing more and more of these technologies. Unfortunately, they're still expensive, but as more and more of them come in, the prices will go down and we'll learn to use them properly.
Yeah, so Alvin, if you may allow me, because this drug discovery, development, and commercialization, I think if supported continuously and properly, I think Edsel we may have a chance to contribute in this area. But before that, I'd like to commend you for bringing up the humanitarian, philanthropic aspect of control of infectious disease because well actually the drug companies they don't want to invest in it too much with the research because well if you have an antibiotic then at some point you get well, so it's not like a lifelong treatment thing, but I think HIV is different, so it's so great to know that there are humanitarian and philanthropic drug companies out there, say from Europe. No? So in our case, in my lab, actually we set up the antiviral assays, both for the active and the latent, and we got it from the United States. And we have some candidates also for gram negative, active compounds. And we were asking for clinical strains, both susceptible and single drug resistant strains from PGH or from his lab. And at some point, we never heard from him again. So maybe we could remind him about it because we are at that stage where we need to test them in single drug resistance. It's very difficult to find a candidate compound for gram negatives, right? So I'm going to, if you don't mind, they seem to be our younger researchers from USM. This must be University of Southern Mindanao. Alvin, this is Yvonne Saliling, and she asks, may I respectfully know if there are limitations of the kit? Can it detect even the early stage of the infection? DOH says that there is this, what they call a window period. The PHLIV is already a carrier, but can still appear as negative during the screening test. So
Yeah.
that,
Yeah, so this is not an HIV detection kit. This is a drug resistance kit which sequences the virus. And the way that it works is that as long as there's a good amount of virus in the blood, usually as long as it's about 1,000 copies, it should be able to do that. Of course, the higher the viral load for somebody who's not on treatment, the deeper we can do in terms of sequencing. Because if your viral load is low, then you're just sequencing the same viruses over and over again. So you're not really going to get a good idea of what the quasi-species looks like. It doesn't really have anything to do with the window period or the carrier period. The window period is really more of an antibody where you can't detect the antibody during the 6 to 8 weeks from the time of infection. Although the newer generation, the fifth generation tests nowadays actually also detect antigen. So even inside the window period, about 20 days. But this test has actually nothing to do with that. It really has more to do with sequencing the virus that is there and tells you if there's pre-existing drug resistance for people who are treatment naive, or if there is post-treatment resistance for people who are failing treatment.
okay, so Alvin, anymore? I see out here, ASEAN has the regulatory uh, harmonized, so an approval in one country can help for the rest. Regulatory harmonization. So looks like we're going to aim high for this. So I recall that just before the pandemic, Secretary Boy de la Peña and Director Dr. Jimmy in a press conference which we attended at the PICC of PCHRD projects. Dr. Irene Villasenor was there. They had said that that dengue kit was being tested in Singapore, okay, because if it passed the test of Singapore, then we would have under the ASEAN harmonization, then we would have the potential market of the ASEAN. So I don't know what happened to that effort. Did Singapore finally give us the results of their testing of the dengue kit of Raul?
I'm not sure if it's a full regulatory filing, Giselle, in Singapore.
Yeah,
because I was
it did not
close with-
happen. Oo.
I'm not sure if they completed the requirements of
Yun na
HSA.
nga.
That's why it's very specific for me. If we can bypass regulatory in the countries with the WHO approval, Edsel, will that
Yes.
allow deployment of your antimicrobial resistance testing?
Yeah, I think so. I mean, FIND, which is their diagnostics
Yeah.
arm. If you notice, that's what they did with
Well, COVID, yeah. Okay,
Yeah.
so I'm
Well,
hoping,
at the
I think
time,
that's the best avenue for you. Thank you.
Yes,
yeah,
sir.
that was that press conference I remember was towards the end of 2019. So we were entering COVID, right, Irene? So we did not. Well, maybe things did not push through. Okay, but it would be great if we could get this invention of Edsel also to move forward. If not in the Philippines, with assistance from
Yeah, actually, the setting completely changed during COVID, Giselle, that having
yes,
Raul's Labinamag utility was greatly reduced by having RT-PCRs everywhere, including Edsel's grandmother, as she said. But definitely, Edsel has more legs to lean on, considering that it can have multi capabilities, not even in a low resource setting. As he indicated, is even more advanced than the current Sanger gold standard. So I think, yes, I'm open to helping out. But again, I think the more important part that's not being address at this point is the regulatory pathway. And if indeed the WHO can allow it to be deployed while you're going through the regulatory filings with the clinical data that's being collected, then I think you will have a much wider reach, because you can involve not only governments but also foundations in order to be able to support your efforts to test worldwide. So I think that's probably my 2 cents in this one. Let's look at the regulatory pathway. If there's no resistance, indeed, Edsel, I think you're off to the races, and we can have a dashboard in terms of the number of patients helped over the coming years. Hopefully, it will be in the millions, like Menchit's drug screening for infants 10 years from now. So, but I think that's what I want your diagnostic kit to be when this is all scaled up.
So
Thank you.
I think that there are two innovation steps here. So a lab in a bag, which is actually designed by Romel Gomez of the University of Maryland. Okay, that's the lamp-based amplification, right? That helps with point of care. But this one of Edsel makes use of Gene Expert for amplification. And then this part that he's using is on the sequencing. Those are two steps, right? Edsel, am I right about this, right? So you're using, yes.
Yeah, we don't use we use Gene Expert for viral loads, ma'am. That's more clinical.
Viral loads. For your amplification, what do you use?
For the amplicons.
Yes. Yes.
Well, RT, because we have to convert the RNA to DNA.
Yes, yes, yes. RT, reverse transcriptase, and then you amplify, and then you sequence, right? Okay. So, well, that's where the innovation is in this case. So the sequencing itself is also now point of care, right?
And near point of care, ma'am. It takes about 2 to 4 hours.
Kahit na. The fact that it's portable, that is a big advancement, right? Okay, so I think with all of these things, clarified to the audience, and I just would like to recognize the presence of our biomedical researchers here. Marge Peña is our Vice President of the PAASE and President-elect, so she will be President of the PAASE. She's in the University of Southern California, and she's actually a cancer researcher, so she knows these protocols as well, which would be related to, of course, infectious and cancer research. But also one of our great mentors in UP Diliman, National Institute of Molecular Biology and Biotechnology is here. This is none other than Professor Amorfina Miren Santos. Okay, who's we were doing research for a long time together, and she trained at the NIH, US NIH as well. And then she did her postdoc there, and then she taught so many of our best and brightest MBB graduates, and some of them are MD PhDs. So the impact of the training, the teaching, I think is very great, and goes through every year out. We try to mentor very good students. So. Alvin, I think you have to. You want to speak now?
No, I think we're good. Although Tito has a question here. Maybe Tito, you can just probably, perhaps the last question that we can entertain.
Actually, it's not a question, Alvin. Dr. Edsel, good morning. Thank you very much for sharing with us your experiences and innovations, etc. Actually, my concern has been voiced very well by Al. It's because our part in industry is to get these things on the ground. Get these things on the ground to benefit millions of people. But again, unless we can bridge that, again, bring our ideas to the hands of people in the barangays, etc., then it's just something in the clouds. Kaya, Al has mentioned it. So anyway, we can help move it again, bring it to the ground. That's where industry will come in. Thank you very much, Doc. Have a great day, everybody.
Thank you.
Po, para magamit po yung science.
Correct.
Yeah. Sige.
So
Giselle, yeah, I think it's already
Grabe, no?
33. Go ahead. You still have some, you know?
Just so we know the numbers, it's really a numbers game about innovators moving to the TRL 8 to 10. That's the commercialization phase, and the numbers are really staggering. So in Indonesia, among 1,000 startups supported by the government, only 5% get to the unicorn phase. So the billion, say in our case, billion peso phase. That's numbers, but 5 unicorns are good. It means that our government has to support research and innovation to a much greater extent, and that's one of the goals of the NIC, because it's in our ASDI
Yeah.
targets, core targets,
Yeah.
which
If
is
I
the,
may, yeah. If I
yes,
may comment, Giselle, about the
yes,
TRL,
yes, Tito.
our TRL measurements seems to lead us nowhere. I say this because many of our TRLs, once we give it to industry to work on, they said, anong TRL 8 ito? TRL 3 lang ito. That's when natutuwa si Al. He knows that. Because that's when industry now puts their hands onto the idea, and marami palang gagawin.
Oo nga,
Sometimes,
may resistance talaga.
hindi resistance. Talaga yung rating natin ng TRL, I think is not right, is not good.
Okay.
So
So,
we
Guys,
better,
TRL is only one marker. That's on the
correct.
technology. We have to
what
assess
I-
investment ready. We have to
Yeah.
assess market readiness. We have to assess regulatory.
Yun nga
And
eh.
so,
That's what I'm saying, that
isa lang yan. That's not the metric for all. So we need to look at all the whole aspects of
so far
commercialization.
kasi,
actually
only
my point is
agency-
then, my point then is that give it to industry as early as possible and let them
Okay.
grapple with it, test it. Kasi in many cases,
Good.
some of our technologies are based on 10-year-old technologies. Pag tiningnan ng industry, naku, luma
Ay.
na ito.
alam natin yan. Oo nga. Tapos, I'm telling you, there is real resistance from industry for high-tech, biotech, biomedical medical
Sige.
research.
Okay.
No? So, in the end, also, the issue of how are you going to, put it on the manufacturing phase, right? Tito. So,
Yeah.
pagka gagawa na ng mga, itong minayo na to is imported, the question also is, of course, your cost is already good, but you would have to bring it down some more, right? So it's
Yeah.
always an issue of the materials that you use, what is imported and what is developed.
Yeah,
by-
Giselle, the main resistance of industry is really about risk. Risk in
Yes,
manufacturing,
absolutely.
risk in regulatory,
Ayan na nga.
risk in
Ayan nga.
clinical use.
Oo.
So unless you de-risk the technology Edsel, we will have big challenges in scale up.
Yeah.
So we need to
Yeah.
remove all those risks.
Yan
Yeah.
talaga
Thank
ang yan talaga
Anyway,
ang
I think, Giselle,
Oo.
this innovation fireside chat series is really the intended venue to have this
Correct. Yeah.
diba,
Yeah.
aired, and so that
Yes.
Giselle, as one of members of the executive member of the NIC, can bring this right at the council, because many of those in the past, especially from our government officials, have no idea really what's really happening on the ground. And I think this platform, this fireside chat, is what we're trying to do across many innovators, startups, and ideas. So we're trying to pick up all those challenges. Tito, you've
Yes,
always
yes.
been reminding us
Yeah.
about this, and this is what has to be really brought to that NIC. And hopefully our team there with Giselle, Tirso, and of course
Rex,
from ELB,
Cruz.
yes, Rex, can really push their thinking of this legislators, of our government, decision makers. And hopefully there will something that will come out of this. And I think that is the good thing is the NIC is chaired by no less than the president himself, co-chaired of course by NEDA. So anyway, I think Giselle, it's already 38 past the hour of 9 here. So I think you have any other And maybe we have to show already the other.
So
Plans?
as I always say, well, at the NIC meetings, I understand in Malacanang, there will be one in February. There's very little time to speak, and the agenda is fixed a month before it has to be submitted. I think to the office of the president weeks before. So it's really great if our R&D and innovation leaders come up with a position paper. So I just had an innovation proposal submitted, and I will be consulting our past presidents of the PAASE on February 2.
Yeah.
So
Okay.
we're going to have a break. We're going to have our last innovator speaker, very, very interesting and important speaker. He's a very, very distinguished OFW, okay, engineer Fred Sylvester. But after that, we're going to have a little consultation with our stakeholders in PAASE. So please help us with your position papers. Al, you should read in writing this, problem of the interface between academia and industry and the regulatory. Right now, the department that is implementing the TRLs would just be the DOST in a systematic way. And if you listen to our inventors, innovators on the entrepreneurial side, not necessarily from the university, sasabihin naman nila that, the TBIs and the accelerators of the DOST have also helped a lot, no? But there are not enough innovators because of these hurdles that Al, I think, and Alvin have identified together with myself. So I think that one is, yes, send us your position papers on any topic related to innovation. Second is identify the innovation hurdles or barriers, because there are many papers like this from industry, but none from this university stage of meron na pero it cannot move on to the next stage. All right, so let me say that we are looking forward to an announcement perhaps of the use of the Innovation Fund. And I understand that maybe we will be conducting or holding nationwide and region-wide innovation competitions to draw out those like, say, Edsel and others we've featured here to compete. And maybe the competition will result in a lot more publicity and some funding, and also my own thinking based on what I have witnessed in Taiwan and other countries. The government actually introduces the innovator to the business community to, almost require the business community to pick up the innovations that, have won awards. Okay, so with that, Alvin, maybe last words from you. Thank you.
As Giselle mentioned, we will soon have a break in this Fireside chat series, but I think Giselle will go through the 17th and the last 18th speaker will go through it and then maybe summarize what we have learned from all of these talks, and share this to everyone so that probably when you do the talk in 2 weeks time, I think you're scheduled to have one. Maybe you can give us an idea of what are this, and this will be those that will be raised in the NIC, and hopefully we can all support this and really convince members of the NIC that there's so much to do as far as innovation in this country is concerned. We all experience many challenges, and we cannot really in fact, I'll remember the COVID. We developed this what's this emergency, but it didn't push through because we were stuck in the clinical testing. We were done with the mechanical testing; it passed all, but clinical testing we're stuck in there. So there's just so many areas.
Actually, that's the position that we're taking. Edsel, maybe you can help us in terms of asking for investigational device or diagnostic exemption for us to do clinical studies on things like diagnostics and drugs on humans. Doctors can do anything they want, but we engineers cannot. Unfortunately, kaya hindi kami pwedeng gumalaw sa tao. Kayo
Correct.
dapat pupunta. But then the doctors don't want to handle it if there's no coverage. And again, it's a question of risk. Why am I going to use Alvin's device if wala namang read for the FDA to say, okay, exempted na yan, do your clinical and come back to us with the data. At least diagnostic is less significant in the risk, so you can do that. But definitely as we move into devices and even to Class Luna's products getting into clinicals, we will have to deal with all this. So a meeting, as I indicated to Ted yesterday, FDA talaga, 3 letters is our biggest challenge right now. And Dr. Sam, we invited him at the NAST last year. Did he show up? No. We have another session now on devices. We're hoping that he does show up, but I don't know who else to approach. That's why I'm already begging Ted on DOH. I don't know if that's still the line of power. Maybe it's the president, Giselle, through NIC, that needs to prod the FDA to be more responsive. Even just meet us in the middle, the way HSA of Singapore does for innovative products, both diagnostics and devices. Yun lang ang appeal ko. I've been doing this for 10 years, guys. I have 7 FDA approval in the US. Pagdating ko dito, wala.
Kaya nga,
You know,
I have to bring this up today because during the pandemic, the medical device that you're working on, PCHRD UP Manila, is the Ginhawa ventilator. See, then I wanted to report on it at the TWG for innovation of the UP system, and so I looked it up, and then I found out that the problems were hindi pa magawa yung clinical trials for it, and then walang manufacturing of the medical device. Whereas Indonesia, during this time, had 5 ventilators that were approved, okay, and they were used. One of them developed by Universitas Indonesia, others developed by companies and other universities. So that's the difference between Indonesia and us. So what's the problem, Al?
We could have easily used, Giselle, the emergency use authorization that was allowed for diagnostics. Giselle, Edsel knows that. And we could have done that, lifted that for ventilators. But luckily we did that because apparently ventilators were killing more people.
Anyway, I think
so may problema talaga sa proseso. Even under UAE, hindi nagawa yun, no? Hindi natin naihabol yung ating local ventilator. Until now.
These are the experiences we had really in the past COVID-19, we all experienced that. And I hope we will have some resolution.
Yes,
correct.
Yeah.
We're attacking it even on the procurement front, Alvin and Giselle and Edsel, definitely emergency procurement that was laid out with USAID Stride, as you know. We're doing it at the NAS level as well as UP system and EDCOM even, to make sure that we have the tools and able to procure them. Edsel, as we scale up your technology, you might need not hundreds but thousands. How are you gonna do it? We can't do it through UP. We can't do it through the USD.
Ginhawa was a project of PCHRD and UP Manila. So I presume that procurement for materials to build it, nagawa naman, pero
actually LaSalle is the one that made the device, Giselle. Ginhawa is actually a LaSalle device to be tested by Bunz Baldos,
so
which didn't have the
university is a good one.
So that it was fraught with definitely risks when it was in development, and that's what I don't want other devices as well as diagnostic to suffer. Especially, we need all hands on deck, multi-university collaborations, and even private sector. But unless you remove the risks associated with the development, no one will come into play and will put up the money as well as help you roll it out and scale up. So
this brings me now to the concept of open innovation, which is being practiced in South Korea, especially in the pharma and biomedical industry. Devices.
But the FDA has to be part of the open innovation. If the FDA is going to be a blocker, we will all be open to risk.
merong nga intellectual property has to be addressed. Pero partnering of UP Manila with DLSU, that is great. Okay, tapos may PCHRT support. Anyway, lots to talk about. So,
Yep.
but at
Okay,
least we're
so-
now aware of the problems. Tito is always pointing out things that we should really be doing things on the ground. We have to address these problems, Tito. No?
Yep.
So
Yep.
hopefully
We have to.
Yes, let us give Dr. Edsel Sapalya really a round
great,
of
round of
Hey.
thank you
applause.
so much, Edsel. Wonderful talk.
Thank you,
We're
Doc.
learning a lot, in our Fireside series. So thank you
Thank
so
you.
much, and
Thank
we really appreciate your sharing
Yeah.
your work with us.
Yes.
So yeah, Fed, are you going to show any more announcements for the next series?
Yeah. Yes.
Okay. All right. Giselle?
Yes, this is my friend Fred Silvestre. So we met at a meeting of the NIC with the Primex that they had subcontracted to study our barriers to innovation. And so Fred was there and he spoke at the open forum, so did I. And I knew immediately that this is the guy who would tell us about the prospects of getting our OFWs. Many of them are highly technical people like Fred, who's an engineer from a poor university. And well, I think that this workforce, this skilled, competent, and expert workforce should be joining us in our advancement towards, well, innovation, S&T-based socioeconomic development. So I look forward very much to the talk of engineer Fred Silvestre. Thank you.
Thank you. So again, we wish to thank everyone who is always in attendance, who joins us every Friday morning. Thank you so much. We'll see you again on Friday next week. And yeah, we look forward to another engaging and productive discussion. Thank you, everyone.
Thank you, everyone.
Good morning, good day. Thank you, Edsel.
Edsel, I have to tell you that
Thank
I have been inviting more people from researchers from UP Manila, including academician Edward Wang, to talk about CBOL, and he's still not accepted my invitation. So maybe you could help me.
Okay.
The lead from your chancellor, who was like one of the first to speak on AI-based simulation for medical education. Also, I think that is so important that we hear from our MD researchers like you and Dr. Wang.
Thank
Okay.
you, everyone. Thank you, our university collaborators. Thank you so much. And those who attended. Thank you,
Yes.
Giselle. Edsel.
Thank you
Thank
so
you.
much, Alvin. It's so nice to know that you're home. For once, you're home.
Yeah.
Okay.
Okay. Thank you.
Bye-bye.
Thank you, everyone.
Thank you so
Bye-bye.
much, Deb, and everyone
Bye.
for
Thank you.
joining us.
Bye-bye.
Bye-bye.